Hello, and welcome to this video on DNA methylation. DNA methylation has recently appeared in social media with posts remembering National DNA Day. This was epitomized by Beatrice the Biologist's cartoon on DNA methylation. DNA methylation is a process of modifying the nucleotide backbone of DNA via the addition of a methyl group. This is called DNA methylation. It is distinct from the mutation of DNA. This is classified as an epigenetic change as it alters the structure of DNA in a reversible, non-nucleotide manner. The DNA saccharide backbone is modified with this molecule, the methyl group being the same as this methyl group. DNA methylation normally acts as a block on DNA transcription through interference of gene promoter sites. This is seen in genome imprinting, aging, and cancer. It performs an important role in imprinting, adaptation, and pathology. In mammalian somatic cells, cytosine guanine sequences are somewhere between 60 and 80% methylated. This may account for the difficulty in cloning differentiated cells. Those genes not methylated seem to be specifically unmethylated. Seen here is the bipolar distribution of methylation in a series of gene sequences. DNA is most extensively methylated on the cytosine or adenine base, but not the thymine or guanine. This mostly targets the highlighted amino acids, excluding many proteins. Interestingly, certain yeast strains arguably have less than 1% of their genome methylated. In other strains, it is highly common where it is tied into the regulation of cell division states. The human genome has an unusually low number of cytosine nucleotides, approximately 21% of the expected prevalence. This is theorized to be a result of the largely methylated cytosine nucleotide in the human genome. Cytosine is methylated in lieu of the alternative thymine. This creates CpG islands. These eventually lead to TpG islands, and this is called TpG island transformation. This shift changes in trinucleotide formation, leading to a change in the amino acid expressed. This occurs as a result of deamination, which is facilitated by the methylation. The previously mentioned change creates this list of non-viable amino acids. Methylation occurs via the site-specific DNA cytosine 5 methyltransferase. This transfers the methyl group from S-adenosine methionine to the C5 position of the cytosine residue. This molecule has been mentioned previously in the autism prevalence video. Deamination is a consequence of hydrolysis reactions. This reaction can be instigated by pH and temperature changes. Deamination can also be increased by exposure to UV radiation. Methylation has been mentioned in previous video on autism. The role it has in autism is unknown, but the role in Rett syndrome has been elucidated. In Rett syndrome, it suppresses the activity of the MECP2 gene. This leads to a series of pathologies. Methylated cytosine accounts for 18 to 30% of all germline and somatic point mutations. This is partly due to the lower efficiency in repairing the nucleotide mismatch. It decreases with age and is associated with an increased risk of cancer, particularly as CpG islands are common causes for tumor suppressor gene disruption, commonly the p53 gene. Seen here is work by Wajed et al. That explored the role of CpG islands in deactivation of the p53 suppressor gene. Methylation is generally seen as not something of an inheritable trait. However, there is one exception to this rule. This is through a process called imprinting. Imprinting silences a gene inherited from a parent. Angelman syndrome is an example of this process. Whilst not an inheritable trait, methylation is largely a tissue-specific process. It is most pronounced in the embryogenesis and the necessary differentiation of tissues for that. The process of methylation controls how the genome is expressed. Through control of expression, various growth factors can be restricted, limited, and stopped as needed. This leads to a one-way, guided determination of what the precursor cells will turn into. It further affects how far and when this will occur. Among the findings of this is the low methylation of the egg and high methylation of the sperm. The effects of methylation are very specific and long-lasting to ensure the development and differentiation occurs over the correct time and in the correct place. Deamination as a result of methylation is repaired as a response. This is initiated by the uracil or thymine N-glycosylase. This is commonly in the form of base pair excision and replacement. Considering where methylation occurs, understanding how it works and what it does seems prudent. The most commonly held theory is that methylation physically prevents the transcription of a gene. This is done by altering the physical shape of the DNA binding site. This disrupts the binding of transcription proteins. The second means is demonstrated by Lennertz. The second method of methylation action is through gene dosing. Methylated DNA attracts proteins involved with deactivated chromatin. These aggregate to form a bundle of chromatin, which has a similar purpose to a locked storage box. It makes it difficult to access and use the contents. The bundles are known as heterochromatin. Based on this, methylation deactivates unwanted genes. As humans age, the genes are gradually turned on in an unnecessary manner. In pathologies, they are inappropriately turned off. Methylation leads to the mutation of cytosine and the increased demand for error checking. Despite this, it is highly conserved as it is a very stable means of regulating gene transcription with a very low cost to maintain. Thank you for watching this video. Please post any questions, comments or suggestions you may have below.